Explainer. Today we're diving into an absolute classic of the laboratory world. We're going to break down the chemistry and the actual mechanism of Fehling's test. It's a foundational chemical analysis, and we're going to see exactly how it works from the ground up. Let's jump right in.
Okay, so picture this. You're standing at a lab bench, and you've got this completely clear, transparent liquid in a glass beaker. I mean, it looks exactly like water. But is it? How do you actually detect invisible, completely dissolved sugars hidden right there in front of you? It's kind of a high-stakes guessing game, right? Because if you guess wrong, you could literally ruin an entire chain of experiments. It's such a fascinating problem, identifying these unseen molecules that give absolutely zero visual clues until you actively force them to.
To solve this little mystery today, we're going to follow a pretty straightforward step-by-step structure. We'll start with detecting invisible sugars, look at the chemical principle, break down reagents A and B, walk through the actual lab procedure, and finally, interpret the results.
All right, part 1, detecting invisible sugars, the classical biochemical test. So, Fehling's test is what we call a classical biochemical method. Honestly, for decades now, it has been the absolute go-to method for biochemists, researchers, students, you name it. But you might be wondering, why has it survived for so long in our modern, super high-tech labs? Well, it really comes down to its incredible reliability, its low cost, and just sheer visual clarity. When you've got an unknown sample and you need to interrogate it to see if certain types of carbohydrates are hiding inside, this is the tool you reach for. You don't always need a massive, expensive mass spectrometer to get a definitive yes or no.
But what exactly is the target here? What are we searching for? We really need to define our suspect. Fehling's test is specifically hunting for what's called a reducing sugar. Now, a reducing sugar is a type of sugar, glucose being the most famous one for sure, that has this really specific chemical ability to act as a reducing agent in biochemical reactions. So they aren't just sweet. Chemically speaking, they're highly active participants, totally ready and willing to just transfer electrons to other molecules.
Moving on to part 2, the chemical principle, redox and copper.
So the entire mechanism here is based on a redox reaction. That basically means reduction and oxidation are happening at the exact same time. And this reaction absolutely requires an alkaline medium to work. Here's the specific chemical interrogation tactic, okay? If reducing sugars are present, they'll literally force electrons onto copper-2 ions, reducing them into copper-1 oxide. It's kind of like a microscopic exchange of power. The sugar sacrifices a piece of its own structure just to hand an electron over to the copper. And the brilliant part of this is the visual payoff. See, while copper-2 in solution is this beautiful deep blue, the very second it accepts that electron, it physically transforms into copper-1 oxide, which forms a heavy brick-red precipitate. So this microscopic electron exchange literally paints the invisible sugar red.
All right, part 3, reagents A and B, the two-part key.
If you're looking right at the lab bench, you'll immediately notice a really stark difference between the two bottles sitting there. On one side, you've got Fehling's solution A. That's an aqueous solution of copper sulfate, which is exactly what provides those crucial blue copper-2 ions we just talked about. Then, on the other side, you have Fehling's solution B, which is alkaline sodium-potassium tartrate. Now, why do we even need solution B? Well, without it, the really strong alkaline environment would instantly cause those copper ions to just clump up as a totally useless hydroxide, dropping right to the bottom of the flask before the sugar even shows up. So the tartrate acts as a chelating agent. It gently holds the copper ions in solution, keeping them perfectly stable and suspended until the exact moment our reducing sugar is introduced.
So here's the absolute crucial point. You cannot just go buy a single bottle of premix Fehling's reagent and let it sit on a shelf. No way. It has to be freshly prepared. You literally have to mix absolutely equal volumes of solution A and solution B right before you plan to use it. If you don't make it fresh, that tartrate we mentioned eventually loses its protective grip, the copper degrades, and your test, well, it simply won't work.
On to part 4, the lab procedure, step-by-step bench work. Okay, so let's walk through the exact practical benchtop instructions. First up, you take exactly 2 milliliters of Fehling A and 2 milliliters of Fehling B, and you combine them into a single test tube. Boom, that creates your fresh, stabilized reagent. Next, you take your unknown sugar solution and put it into a completely separate second test tube. Then you place both of these test tubes into a heated water bath. You just let them sit for a few minutes so they both reach the exact same optimal temperature. Now, why heat them separately first? Well, heating them individually ensures that the moment they finally touch, the thermodynamic reaction is totally uniform and instantaneous. It prevents any of those degraded or messy intermediate reactions from happening. Finally, you take the heated test tubes, mix the sugar solution into the reagent test tube, and you watch closely.
Finally,
Part 5, Interpreting the Results, the Final Confession.
So the results of this test are just remarkably definitive. If you mix the hot solutions and the liquid just stays this clear, deep blue color, that's a negative result. There's no reducing sugar present, and the copper just remains completely unchanged. However, if the solution suddenly shifts and drops out this heavy, opaque, brick red precipitate, that is a definitive positive. The copper has been successfully reduced. You've basically caught the target molecule red-handed.
Now, before we wrap up our benchwork today, we absolutely have to talk about the golden rule of safety and precision for this protocol. Always prepare the reagent fresh and avoid overheating. Boiling it too aggressively or, using old premixed solutions can actually cause the copper to precipitate all on its own. And that gives you a false positive, completely ruining your hard work and sending your entire analysis in the wrong direction. In biochemistry, precision really is everything.
Fehling's test proves that with the right combination of logic, structure, and chemistry, we can actually make the invisible completely visible. It's just a highly reliable, beautifully simple method for detecting reducing sugars. But it really leaves us with an exciting thought to chew on. If a simple mix of copper and a hot water bath can reveal a hidden sugar, what other invisible biochemical secrets are waiting to be uncovered in your next lab sample? Keep questioning, keep learning, and I'll catch you in the next explainer.